You and me. We all started from this one little zygote, which resulted from the fusion of a sperm and an egg. Yet, how does the division of this one little zygote resulted in trillions of cells that make us up? We have approximately 200 different types of cells, varying from our muscle cells to blood cells to neurons. This is when epigenetic comes into play. So, what is epigenetic? Epigenetic is the changes in the organism caused by small chemical tags on the genetic material, but not the alteration of the DNA sequence itself. We all know that the nucleus of the cell contains our DNA, or the genetic material. The DNA strand is coiled around proteins called histone to form chromatin and eventually chromosome. A section of the DNA that codes for a protein is called gene. If genes are not expressed, proteins cannot be made. So, these epigenetic tags on the genetic material can switch genes on or off. All the cells in our body started as the same kind of cell called an embryonic stem cell, like these books. But when paper clips attached to different sections of the book, it switched certain parts off, leading to two different stories, like how stem cells are differentiated into specialized cells when epigenetic tags switch off different sections of the human genome. One way of switching genes on or off is by DNA methylation. DNA in our nucleus is surrounded by small molecules. Out of them, a methyl group with the help of an enzyme called DNA methyltransferase attached to the cytosine base in the promoter region of the DNA. This switches the gene off. The RNA polymerase cannot get access to the gene, and therefore the necessary protein is not made. That is one way our brain cells remain as they are and not turn into a skin cell or a muscle cell, because that would be gross. On the other hand, in histone modification, molecules such as methylphosphorus acetal come and tack onto the histone protein, making the DNA strand around it to become tighter or looser, thus easier or harder to access. Gordon Warnington described this differentiation process using his epigenetic landscape, like how it is hard for the ball at the bottom to roll up again. Adult differentiated cells cannot go back to embryonic stem cells. But when scientists inserted certain essential factors, these adult differentiated cells actually become stem cells again. These cells are called induced pluripotent stem cells, or iPS cells. iPS cells could beat many diseases and even be used for organ transplant. Scientists could store the iPS cells of the patient, guide them to become the desired differentiated cell, and replace the damaged cell in the patient's body without immune rejection. If our DNA is the script, then we are like movies. Our epigenetic tags are like directors. They only give out certain parts of the script to different actors, ourselves, which act out to make a great movie, or us.